Professor Maxime Descoteaux is going to give the first lecture of this semester. And Maxime is professor in computer science since 2009 at the Science Faculty of Sherbrooke University, and the scientific advisor or at IMECA. IMECA Solution is a company that he created with some colleagues. In 2021, he became a member of the College of the Royal Society of Canada. And he is the founder and the director of Sherbrooke Connectivity Imaging Laboratory, where there are 10 to 15 graduate students and medical students and postdocs. He holds the research chair in neuroinformatics, and his research focuses on brain connectivity from state-of-the-art diffusion MRI, multimodal acquisition, reconstruction, tractography, and processing and visualization. So the aim of his lab is to better understand the structural connectivity, so anatomical connectivity, develop a novel tractography methods, so to follow the tract within the brain, and to validate them. And this is very important. It's not only modeling, but validating tools. And this is because we are developing tools to make them available for research and clinic. So this need to be validated and used as for brain mapping and connectomics applications. So his lecture today is on mapping brain white matter with diffusion MRI tractography. So thank you very much, Maxime, for accepting our invitation.
Thank you. Yeah, I got the micro. Great. Yeah, there's something. Let's see. Il y a une souris à quelque part? Non, non, mais c'est parce qu'il y a quelque chose qui bloque l'écran. Quand tu as parti le recording, j'ai plus ma... Je pense que c'est la souris. Ouais.
Donc, cette souris marche.
Il faudrait l'amener ici, en fait. Non, parce que c'était ici, regarde.
Parce que là, vous avez pas l'écran, c'est ça?
J'ai perdu le contrôle.
Sur votre écran, ici, là?
Non. De côté, peut-être? Non.
C'est bizarre, parce que...
On peut utiliser ça, non?
Bah, ça, c'est le clavier de l'ordinateur.
Ah, là, c'est bon. J'attends un petit peu.
C'est bon, là?
Ben, on dirait que je vois ma souris, mais... Ah, OK, là, j'ai le contrôle, là. OK, c'est bon.
OK.
Ouais, OK. Bon, il y a ça dans...
Bah, ça, vous pouvez cliquer sur...
Ouais, mais je vois pas la souris.
Ah.
Mais tant pis, c'est pas grave. All right. Good morning, everyone. I have a chalk also. This is a class, so don't hesitate to stop me. There are very few equations, even though we're the Centre de Recherche Mathématique. I really want you to try to understand what's diffusion MRI, get an intuition for it, tractography. And I'm also teaching this class as if you had data with you. So you can rewatch the recording and walk through my slides and my do's and don'ts of diffusion MRI and tractography if you ever want to integrate this modality into your research, okay? So like Habib said, we often talk about neurons and the huge amount of neurons we have in the brain, on the order of 85 billion neurons. We rarely talk about the underlying white matter, the actual axons connecting these neurons together. And we have an estimation that we have roughly 160,000 kilometers of axons underneath these neurons doing the wiring diagram of the brain. So quite humongous in number, and all crammed in our small head. So today's task is to try to do a virtual reconstruction of these 160,000 kilometers of axons. Of course, there's a notion of scale, right? I'm not tracking individual axons. This is not me. We're going to work with MRI and diffusion MRI. So we're on the other end of the spectrum at the macroscopic scale. But if you're in Frédéric Lesage's lab, imaging 2 photons or microscopy of individual axons, well, you can see that these numbers are monstrous. And to reconstruct a whole brain connectivity matrix of all these axons and neurons will be a challenge for humanity for the next hundreds of years. So at my scale, at our scale, we're talking about macro connectivity. Luckily, as you're going to see today, these axons tend to organize themselves in groups, in families. And here I photoshopped a few in red, white, yellow. So the motor connections, the frontal connections, the visual tracts here in blue, they tend to group in large range connections from a few centimeters to almost 2 meters. I have axons going from my motor cortex all the way down to my feet, 2 meters long. And as they come to my spinal cord, out of the spinal cord, they group together and they come and map the whole motor homunculus. So these individual axons at the micron level group themselves together, form families that we can actually pick up at the millimeter level with MRI. And that's what I want to show you today. How can we infer this microstructure, even though we have measurements at the macrostructure level? So, basic principles. MRI, we're going to use the same machine. 1.5 Tesla, 3 Tesla. The same machine. We're just going to play with imaging gradients, and I'm not going to focus on the physics today. Again, just try to give you an intuition about the process. But given our MRI machine, we're going to acquire a bunch of images in space. Maybe most of you here are familiar with functional measurements, which is acquiring measurements in time. Here, we're going to acquire different images in space. We're going to also use maybe a myelin contrast, an anatomy, a T1 contrast. We're going to put all these things together to reconstruct the macroscopic connectome, i.e. the set of all possible connections at the macro scale. And in this image, you have roughly millions of lines. They're not axons, right? Each line is a family of axons grouped together, connecting 2 gray matter regions together at the macroscopic scale. All right, so diffusion, random walk. What you're seeing here in the video is random walk of water molecule. It's a little simulation. Red and green water molecules just randomly walking together on a 2D plane, bouncing around, randomly walking. So this is random walk. Thanks to Albert Einstein, we have the equations of free diffusion here. There are no obstacles except the blue square here. So these random molecules are just walking around freely. If we introduce obstacles in there, right? Cells, membranes, structure, this will affect the random walk of water molecule. This is at the heart of our diffusion signal. And a key concept here that I want you to understand is that these molecules have a speed here, right? Millimeters squared per second. And I'm not giving you the equation, but you can imagine that these molecules have a speed. They are randomly walking around at a certain speed. So if they're in blood, if they're in water, if they're in bone, if they're in a piece of steak or white matter, that speed is going to be different. And the key word here is the apparent part. Okay, this diffusion coefficient will be different depending on the medium. And actually, if you go to Wikipedia and you type apparent diffusion coefficient, you'll have a whole table of what is the ground truth apparent diffusion coefficient of aqueous water. Water at 37 degrees, water at 0 degrees, blood. And so this is quantitative. And this is what we're going to try to measure with MRI. This was the revolution of diffusion MRI, is that we can use an MRI machine to calculate what is the apparent diffusion coefficient of water in CSF, in cerebral spinal fluid. Okay? And so let's take a video. Again, I'm very educational here. Imagine we're filming a party in front of UDM, and we let people freely drink, speak. You can see this as random walk, right? But if we move to the party to St. Catherine Street, the Alouettes won last night. Yay! Look at the party now. Right? It's constrained by the structure. If you move to St. Catherine Street, suddenly the random walk is anisotropic. If you move to the party to the intersection between St. Catherine and University, is that a crossing? Yeah. You can suddenly see that the party, as at the beginning, looked isotropic, looked random, but suddenly that's a very strong concept. Right? 3 phenomenons. And now I can remove the Google map, remove the buildings, and just say, okay, these are my measurements now. If this is what I'm measuring, I can infer that here I was in a 2 streets crossing. Here I was in a single street. Here, oh, this is kind of weird, right? Because I let them party for too long. But at the beginning, it was very equally likely everywhere. This is diffusion MRI. This is what we're going to measure with your water molecule. It's just that suddenly your water molecules are in your brain, they're trapped in your tissues, or they're free in CSF or blood. And that's going to allow us to make measurements, to make images, because the deviation from free will create a contrast. So, a human brain that we see from top, and I'm going to zoom into a little section here. So imagine you have tubes. These are my images of axons. Axons are like tubes. And I'm slicing them, putting them under the microscope, or like this. So essentially you have water trapped inside axons. They're trapped, but they're bouncing around, right? You have water outside axons in the extracellular space. And this is a space that's a very hot topic right now, because this space is filled with astrocytes, oligodendrocytes, capillaries, and we don't really know what's going on here, but water molecules are a little bit more free there than inside the axons. Fair enough. And if you look in the other dimension, because these axons are like spaghetti or like structure, it's easier for the water molecule to diffuse along axons than across them. So this is happening in 3D. Okay? So basically, we're going to use the MRI machine and the different contrasts to measure the apparent diffusion coefficient along different directions. Right? I'm going to ask the MRI to tell me, okay, when you're measuring in X in this direction of the corpus callosum, what is the speed of water molecules? And what is the speed of water molecules in Z and Y? And we're going to do this in 3 dimensions. Put this in an algorithm and try to reconstruct the 3-dimensional apparent diffusion coefficient. What made diffusion weighted imaging really, really popular was at the turn of the 1990s, when suddenly they realized that when you have a stroke, an accident cerebrovasculaire, you go to the emergency room, you take the standard T2 weighted images, it can take up to 24 hours to 7 days to see something of the injury, of the stroke. Whereas when you watch diffusion coefficients, the apparent diffusion coefficient, right after the injury, you right away see a drop in signal or an enhancement in the diffusion weighted images. This was the killer app for radiology. No modeling, no quantification. You just put somebody in the MRI for 2 minutes, you see the contrast, and you right away take different scenarios to treat them. This put diffusion on the map. This was invented by Denis Lebihan, who's now in France at Neurospin in Paris. But as of today, it's still the killer application. I'm going to show you a little bit about tractography and how that's making its way to neurosurgical application. But just based on this phenomenon, basically, as soon as your water molecules are affected, diffusion picks it up. My analogy to that is, imagine you had a satellite filming our streets, and the water molecule are cars driving around, right? As soon as you have a roadblock, an accident, maybe you don't know what's happening, but all the cars are driving around a phenomenon, an accident, an injury. Well, even though you have a satellite measuring big pixels on Earth, you can still pick it up because all the cars are doing the same thing. That's what's happening here. So, but how can we go beyond this simple apparent diffusion coefficient? Let me show you in one slide the potential of diffusion MRI. Take this healthy bundle of axons, and remember the colors here. Green is for axons. We'll hear a bit today about the G ratio, but the myelin around axon is in purple. And we have different cell types, microglia, astrocytes, oligodendrocytes around the axons. And this is a healthy piece of white matter. And these are different scenarios that could happen with disease, with stroke, with neurodegenerative diseases. You could have microglia activation. You can see these cells now are activated, they're blown up, and you have edema, an increase in water around the tissue. Or in this case, you can see 1, 2, 3 axons instead of 4 or 5. I'm losing axons, I'm losing myelin. This could be the scenario of a demyelination disease like multiple sclerosis. Well, let's for fun do the microscopy game. So suppose we put these tissue under the microscope, right? We put these tubes, we slice them. So this in mathematics, we love this, a little simple model, right? Green tubes, water around them. So we have intracellular water, extracellular water, right? So if I have a machine that can estimate the area of green and the area of blue, I have a measurement of intra versus extracellular water. In reality, well, it's not that different. Okay, here you can see the light of axons is in gray. The myelin is in dark, and here I Photoshop the result. So you can see the extracellular water in blue, the green are the axons, and the myelin sheath in purple. So if I have good models and good data, I can estimate fiber density, how much axon in my pixel, how much myelin in my pixel, right? And how much free water in my pixel. If this works, we have biomarkers of disease, right? We have biomarkers of treatment also. If you have a remyelinating drug, or if you think your drug can create new neurons, new axons, suddenly you need to measure fiber density. If you have remyelination drug, you need to measure the purple. If you have something against inflammation, you know, the future Tylenol of the brain, well, in principle, it should reduce the extracellular water. It should remove the edema. And so that's the holy grail of diffusion MRI. We're not there yet, but as Abib was saying, we founded a company 10 years ago, and one of the goals of the company is to try to bring these markers to the market for the future drug developments of brain disease. So back to our images, back to MRI. This is a T1 weighted image, and this is a myelin map. Kristen Tardif is coming on Friday. She's going to teach you all about reconstructing myelin maps. What do I want to show you here? A bit of anatomy. So first, gray matter is great. You see the whole gray matter ribbon. And white matter is white. But where are my 160,000 kilometers of axons in there? Where are they? Where's the contrast? None. No contrast, right? I cannot do tractography on this. All I could potentially do here is say, I'll segment the white matter. I'll take a volume. How much white matter is there? And that can be useful. But that's pretty much where volumetric T1 analysis stops for white matter imaging. If you want to go into the white matter, you need contrast here. You need to know where are my axons, and most importantly, in which orientations, right? Axons are roads. We need the 3D Google map. And that's where we need diffusion. So again, no equations, but I just want to show you that if I go in the MRI, and I used to do this as a student, I used to put a piece of celery or a piece of steak in the small animal scanner, take an image in X, then put I used to rotate the specimen, put the celery in Y, and then put it in a little tube and put it in Z. We don't have to do that anymore. The MRI does it for us. I can say, acquire images in X, Y, and Z. And look, you don't need a PhD in diffusion MRI to appreciate the contrast. It's ugly. I'll give you that. It's low resolution. It's pixelized. And you don't know anatomy here, but remember when I was saying my motor tracks come from my spinal cord all the way up to my motor cortex. Look at this. If I measure in Z, the image here is dark. This is dark. But if I measure in X, this is bright. It's the same brain. It hasn't moved in the MRI. What's moved is the direction at which I'm acquiring the images. That's as an image processor. So the radiologists are screwed here. They're like, what do we do with this? This is where the mathematics and the engineers or the computer science comes in and says, okay, let's put that into a rotation invariant model, right? If I want to reconstruct at each pixel the orientation of my structure, I need to put this in a model to get this orientation. And that's why we're moving past the concept of just speed, apparent diffusion coefficient, which was just a number, right, in stroke. No matter what the orientation, there's a stroke, you have a big blob, you're sick. Here, we're trying to refine the methodology to get at each voxel the orientations of our axons. And the next step is the integrity of our axons, right? The amount of myelin, the amount of axons, the amount of water. And so, little sexy video. We're going to acquire a bunch of images and essentially be able to be robust to crossing fibers as well. Remember my first video about the parties on the streets? Because our voxels are so big, right? We're at the MRI resolution. This is on the order of millimeters, and these axons on the order of microns. We have crossing fibers everywhere. Fibers coming together, but also crisscrossing and traversing to reach cortex. 160,000 kilometers of axons. And so we're going to have water outside axons, the extracellular water. We're going to have water inside axons. And on top of that, axons may be crossing or kissing, right? So how do we do it? I'm doing for time. Okay. So here's the part of the course where wherever I put a star, this is like my recommendation and my consensus. It's been 20 years that this field has been progressing. So if you watch the video back again and you have data yourself, go back to the stars. And the warnings, it's the challenges and something to discuss for research and what I'm going to discuss maybe after the break today. Well, first thing I always tell students, and especially computer scientists, all my students in my lab are geeks that like to program. They never look at their images. Never. They just want to program and write code. It's like, look at your images. Often, the crappy images your eye will detect easily. Okay. First thing you need to be aware, depending on your MRI, not all MRI can measure the directions properly. So the first thing I tell students is check how were your directions acquired by the machine. Visualize your gradients, like these colors I was putting on my MRI machine. And this is just a simple example, but with Simon Duchesne and Louis Collins, we have a project with 30 sites across Canada. We ask all the sites to run the same sequence. And we got crap back, okay? It's not all scanners, unfortunately. These are still kind of research machines. This diffusion type of acquisition is still a research type acquisition. So it's easy to have bad data. And what happens when you have that is that DTI or diffusion MRI is very unreliable. Things really change across Siemens sites. And this is the same person, by the way. Simon Duchesne traveled the whole Canada. It's the same brain in all these sites. So we have Simon Duchesne 108 times across 30 sites in Canada. This is downloadable. He has fMRI data. He has T1 weighted data. So if you're developing new code and you want to say, is my hippocampus volume segmentation robust? Grab this data and check if Simon's hippocampus is robust. Of course, he's aging in time, though. There's probably a 5-year age gap between the first scan and the last scan, but I was shocked to see this when I started creating maps. So I would say to students now, DTI and diffusion MRI is still quite noisy and unreliable. So be careful. We're becoming better and better, but first thing to do is watch your images and look at the gradient schemes to check if at least the directions where the diffusion was measured are the same. Diffusion is like a movie in time, but it's not in time, it's in space. Remember the 3 images I showed you in X, Y, Z? Now I'm navigating across my 60 or so diffusion weighted images, or more, 100. What do we see here? It's striking. So it's about a 10-minute acquisition. We see movement, right? We see a bit of motion. It's impossible to stay still for 10 minutes in the machine. So you'll see an important part of the pipeline, of the tractography pipeline, is to correct for motion. This is a healthy brain. Look at this crap here. What is this? And this part of the signal is, why is it always more bright? Or the top of the brain here is more bright than the bottom. Okay, this is real life science. This is where you cannot, it's very hard to simulate this. These are artifacts of the acquisition. And I could spend a full hour teaching diffusion and the physics of diffusion and why this is, I mean, the cool thing is that this is feasible in 10 minutes. Why is it feasible? We need acceleration. We need to go quickly. We're acquiring 3D images every 5 seconds in space. And because we're going fast, we have what's it called here, susceptibility artifacts. We go from air to the sinuses to brain, and suddenly there's a collapse in the signal. There's a huge change in magnetic field locally, and we have these drops. So we need to correct for that. There's noise, there's motion, there's eddy currents at the boundaries. So lots of problems that, luckily, these were big issues when I was a PhD student. Now, 20 years later, these are kind of solved. So that's cool. We can really get nice, high quality data. But I say inspect, run these videos. Now we have tools that can create HTML files. You can just look at them in the browser. And if you have a missing slice or huge artifacts, just throw the data away. At the era of the UK Biobank, where we have 50,000 such images, Human Connectome project with thousands of images, I think it's important to do proper quality assurance. Same on the T1. T1 rarely fails, but I think it's also important to check your anatomical image. All right, so let's start processing. When you watch this tool again, you can go back. These are the most famous tools out there if you start processing diffusion. This is our own on top here, using latest, I would say, pipelining languages, Nextflow, containers. So I'm biased. This is a good tool, but I mean, this tool essentially packages together the best of all these tools. So we're not reinventing the wheel. MRTrix, DiPy is open source Python. This is C++. FSL is very famous, of course, from Oxford. This is from NIH, Explore DTI. So what QSI Prep and what Tractoflow is trying to do is package together the best of these tools to correct all the artifacts, the noise. And so I'm going to walk you through. And luckily, I think as a community, we're kind of reaching a consensus. We used to fight about my tools better than your tools and blah, 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 blah. That's over. It's like, we're not talking about tools, we're talking about steps, right? Do denoise your data, do motion correction. I don't care which tool you use, but do it. And that creates better high quality data. So, Tractoflow. So this is the paper. The link, the GitHub, there's a read the docs. So it's quite convenient. Most of our collaborators in Montreal, McGill, Concordia now use it in Europe. So we have a big user base now, and we try to push this forward. The scary part is from raw data to the tractography, it's about 26 steps. And each step is probably 2 or 3 parameters. So yeah, as soon as we acquire a new data set, like a mouse or a macaque, we have to redo everything. All these steps, keep in mind, are tuned, and the defaults are for a 30-year-old healthy student brain or human connectome brain. So if you throw at Tractoflow an Alzheimer's disease brain or an MS patient with lesions, that's going to talk about later in my research talk, but be careful. Most of our neuroimaging tools out there are developed on healthy, young brains. So challenge the steps, and if you understand what the steps does, you can actually change the parameters yourself. So first step, denoising. So the light is not great, but you saw in my images quite a lot of noise. But the beauty about diffusion MRI is you have tons of redundancy, right? We're measuring the same structure under 100 different directions. So it's very easy to put this in there's so many tools out there, but something that exploits structure, neighborhood spatially, but also angularly, to denoise without blurring the edges. So it's very nice now that we have strong tools to remove noise, and that makes us able to push the resolution. We can actually deal with noisier data than we used to and try to have smaller pixels here, because our denoising algorithms can sort of enhance SNR. Then I was talking about eddy currents, motion, right? When you saw my video, you could see the edges of the brain, the frontal lobe. I haven't talked about these colors yet. We're going to see what these colors mean yet, but you can imagine that this versus this is quite different. That's 20 years of PhDs and tools and software engineers nicely packaged in FSL or turquoise. These are not tools from my lab, but they're, you can see the star here. This is a must. And there is consensus on this. Outlier rejection. I love these images, but if you start QCing your input data, you're going to see lots of crap like that. Lines, slice drops, artifacts. And funny enough, don't toss the data away yet. FSL now has options, slice drop correction. So by default in tractofluors, for instance, we turn on all these options because they're critical for diffusion MRI. It adds a little time, but it saves some data sets. Again, this exploits the redundancy because if suddenly the direction just beside doesn't have that slice drop, you can actually use the neighborhood angularly and spatially to complete and paint the missing data. By the way, everything I'm showing today, slowly and not surely, I'm laughing and you see where I'm going with this. All these bricks are suddenly replaced by a deep learning process, right? It's not surprising. I'm not a big fan because all these deep learning techniques are based on these tools, are trained on these tools. They're not trained on ground truth. They're trained on just other tools. So they're just replicating tools, but they go a little bit faster. And I can see the day in a few years where all these steps will be replaced probably by a deep neural net. Distortion correction in the frontal lobe, in the back. And I'm just going to show you a video here. This is before and after. Whoops, sorry. You see this brainstem here? And I purposefully put the anatomy behind. The T1 weighted image is not distorted. But the diffusion image, if you don't do anything, is distorted. Look at the frontal lobe. Here it's stretched. The corpus callosum is not even on top of the real corpus callosum. The brainstem is, this is a non-linear deformation because we're acquiring data quickly. And since in this summer school, we want to infer brain functions, and Abib stressed the notion of anatomy, if we're going to put all these things together, we need a good registration with anatomy, and with the surface, and with the cortex, and with the deep structures, and with the brainstem. So this is probably the most critical step in diffusion. But it's doable, and we have good tools that do it now. So in this paper, and I'm going to come back to it in my research talk, in 2017, this is a 100 co-author paper in Nature Communication. We did just a consensus paper on the challenges of reconstructing tractography in the human brain. And what I want to stress today is that we showed quantitatively that these steps do improve the connectome. We proved it. Out of all these, and this was a challenge and a competition, out of all the submissions of tractography where they didn't correct eddy or distortions or denoise, the resulting connectome was worse than if you do apply these techniques. So they're really important, especially if you're going all the way down to the connectomics. All right. Any questions so far? Because that's a lot of material. No? Diffusion, basics? No? Okay. All right, so let's go a little bit beyond the actual raw images. Everything I showed so far was the introduction and also how to correct the raw diffusion weighted imaging. Now let's start modeling a little bit to start getting at orientations and integrity of the microstructure and to get at tractography. So DTI, very famous acronym, means diffusion tensor imaging. Okay, and the word tensor here is the model. We're going to use a second order tensor. And I'm not going to do the math, but which is nothing more than a Gaussian in 2D, a normal distribution. And so if you acquire at least 6 images, before I had only 3, but 1, 2, 3, 4, 5, 6. I think if I all give you 10 minutes to think about what would require a system to be rotationally invariant, that is to, at every voxel, to estimate what is the distribution of water molecules inside a voxel. This is actually a 2D Gaussian with a principal direction, right? And a size, a length for each direction. So we need the principal direction, and so maybe I should draw it. Right, so with these sizes plus the orientation, that's 6 unknowns. And this can be anything between, right, a perfect party randomly walking around in 3D. This is the sphere where lambda 1, lambda 2, lambda 3 are equal, all the way down to a party onto a street with a certain anisotropy, and this can go up to like something very sharp, right? This is DTI. So in theory, if you do the math, you only need 6 images. In practice, you need at least 12 because this is noisy, and a good rule of thumb is twice the amount of measurements than unknowns. And now, because things go so fast that by default, most MRI just by default do it 30 directions or so. Okay, so this is DTI. Peter Basser is at the NIH. He got the ISMRM gold medal for inventing the DTI in 1994. The inventor of diffusion weighted images, Denis Lebihan, which I was talking about, was his last author on this paper. So Denis Lebihan was also at the NIH back then. And this is 10,000 plus at least now. And so this is the model. So the diffusion tensor with 6 unknown can be something between a sphere and an ellipsoid. And we'll get back to, and I'm sure people are already saying, oh, Max, you talked about crossing fibers earlier. It's coming. This is not a perfect model. It's a good step forward. And remember that there was a time before we had parallel imaging and before we had acceleration techniques where DTI was long to acquire. So acquiring more than 12 images was already a challenge. So we needed something better than just apparent diffusion coefficient, and DTI came along, which was so with this model, we can model anything between a cigar, a disk, right? If lambda 1 and lambda 2 or 2 lambdas are equal, this is a disk, a frisbee, and a soccer ball, a sphere. And this just there is quite useful. We can actually create an images of where do water molecules are spherical, where are they cigar-like, and where are they disc-like? Because these will be crossing fibers, right? And suddenly we can create an image. And for years, that's what we were doing. Right? So these are the speeds and our different scenarios. So water, CSF, blood is spherical. crossing fibers, green, and red are cigars. But this is far from perfect. And then we invented fractional anisotropy, FA. And you can see FA goes from 0 when you're a perfect sphere to 1 when you're a perfect needle. Okay? But the problem with FA is that when you go at FA at around 0.3, 0.4, 0.5, look at the different shapes that the diffusion tensor can take with the same FA. So if you try to summarize the anisotropy here in one number, it can be potentially dangerous. Right here you have like a Smarty, which looks like a crossing fiber, or like a fat cigar, which could look like a single fiber population. So that's why we have big biases and big question marks when we just use FA. But FA is probably the most used feature of DTI. You see FA everywhere in the papers. Why? Because it works. I mean, it lights up the white matter. And if you have disease, or if there's something wrong with your white matter, FA goes down very often. So it's been very powerful for that. It's just that we have to be very careful of how we interpret FA, because we're hiding information in FA. But you can see in my message, I'm a big believer of all models are wrong, but some are useful. Very famous statistic quote. Even my more advanced models are wrong. They don't take into account astrocytes and oligodendrocytes and extracellular space properly. So FA and DTI is wrong. It doesn't model well crossing fibers, but it's useful. And the question is for us as scientists, when we use FA, let's challenge the interpretation. Let's look beyond FA to interpret when things go down or when FA is in the range of these values here. Remember my talk and say, oh yeah, this could be different scenarios in the biology here. So let's be critical, and we have tools to actually zoom in and be a little bit more informed about what's going on with the tissue. All right, so FA lights up the whole white matter. So yeah, from something like 1 to 0. But I mean, this person is not sick, or there's not a tumor here, right? Or here. This is in the middle of the deep white matter. And suddenly the FA is like 0.1 or 0.2. Because two large highways with FA of 0.8 are actually crossing there. And it leads to an FA of 0.2 or 0.1. This is not pathology. But this looks like an MS lesion. If you look at multiple sclerosis, a lesion would actually have this signal. So we need something a little bit better. And suddenly you see my colors. These beautiful FA colors, these are the colored FA. It's the same feature as FA, where I added a color in the RGB world. So I'm coding the principal eigenvector, E1 here. In the RGB channel. So this is a different data type. And it's actually a 4-dimensional images where the intensity is the FA, but the color is the XYZ component of the principal eigenvector. And suddenly, ooh, yeah, blue is the motor track, green coming out at you. And suddenly, in the late 90s, people started building atlases with these maps. And whole books like Gray's Anatomy labeling, okay, this is the cortical spinal tract, this is corpus callosum, this is arcuate fasciculus, this is the memory tract, this is the vision tract, without tractography, just with images. And as a bonus, we go back to our diffusivities. These mean radial axial diffusivity, I should have put units here, but these are in millimeters squared per second. These are speed. This is as a speed. This is the speed at which the diffusion of water molecule in the voxel diffuses in the axial direction or the perpendicular direction. You see the equation right here? Perpendicular is radial, lambda 2 plus lambda 3 divided by 2. It's the average speed of water molecules perpendicular to the axons. So think about disease again. DTI is wrong, but it's useful. Right? So if you have here water molecules here, it's not rocket science. It's stupid, but here the axial diffusivity and the radial diffusivity are going down. Sorry, axial diffusivity is going up. It's actually easier to, well, I can go there. I should have, right? It's easier for water molecule. Now the axons are, there's less axons and they seem, maybe they're aging, these axons. So there are more water in the axial diffusivity direction and more water in the radial diffusivity. Right? It's easier for the water molecule to bounce left, right here. So for a lot of people, like less myelin, more radial diffusivity. If you have less myelin here around the axons, you have less obstacle. It's easier to traverse radially. You have at least thousands of papers showing you that radial diffusivity goes up with demyelination. Axial diffusivity goes down with age as axons atrophy. And so you can see how this is powerful, but also dangerous in a sense, right? Because we don't know what's going on with the tissue unless we put it under the microscope. And we're making big claims with this simple Gaussian, with this simple tensor. And so let's be at least critical about these findings. Let's try to replicate them. Now there's no excuse of database, but when we started 20 years ago, having 20 subjects was big. This is no longer the case. We can go validate our findings in the UK Biobank. We can use different databases to replicate our results. We can do simulations to try to reproduce these phenomenons with tubes and axons and cells. And so this is where we're at as a community. But yeah, DTI is here to stay. No matter what we improve. And I don't know if you realize I would change my message, but I used to be against DTI. DTI is wrong. We need to model this and be better than DTI. DTI is shit. No, DTI is great. DTI is great. It's super useful. And if doctors barely use this now. So before we go to the next step, let's understand what works well here. And when it doesn't work well, let's provide better tools to interpret what's going on. One thing, if you start doing DTI, that's very useful is the residual map. I think most of you probably know what the residual map is, but let me just remind you, if you have a cloud of points here in 2D and you draw a line, this is exactly what DTI is doing, but in 6 dimensions. We're drawing a tensor across 12 images that we're acquiring. And the residual is just the sum of squares of the distance to the pit, right? So we can estimate how far from the measured data is our estimated model. This is a residual. When we do this with images, and I think this is a message for probably fMRI as well, or any technique, visualize these residuals. Suddenly where you had artifacts or maybe something you missed in your QC, or you have a big outlier, this lights up in the residual maps. So in TractoFlow, we always output the residual and we show it to you in an HTML report. So that's the holy grail of residual. It never happens. Like a perfectly flat image with no structure. There's always a bit of structure. But if suddenly, like I wouldn't have seen this ring on the FA. It's very hard to pick up. But there's actually an image artifact, a ringing artifact from repliement d'image inside the image, and you can see this big ring. Will this affect my analysis? Are these tracks important? And you need to think if you remove this subject or you keep it, but this is a big artifact that may be polluting your radial axial diffusivities. Yeah, I'm going to skip this and at least get to the limitation of the tensor. And you can see how passionate I am about this, but I did my PhD in this, that's why. This was the beginning of my PhD. It's like, okay, the tensor doesn't work well when we have these crossing configuration. At the time, we didn't even know if this was a big problem or not. Did this happen often or not so often? In fact, this happens between 2/3 to 90% of the time at the image resolution that we're at. So it's a big problem, especially for tractography. Because if you're not modeling these crossing fibers, basically you're just tracking the center line of tracks and you're pushed away from structures. It's also, I already said a little bit about this, but be careful about FA changes. And we're going to get back to this later in the talk. But imagine that you actually had something like this here. And only one of the two fascicle is deteriorating. The other one is fine. FA is going to break here. And it's going to show you a profile that might lead you to an interpretation that's wrong because you haven't taken care of this healthy tensor here. So we're going to see how we can do this better. But so crossing fibers are everywhere. 160,000 kilometers of axons packed. If you actually looked at tractography, you validate, and there's a beautiful paper that says, even if you go to smaller pixels, smaller voxel, crossing fibers are still everywhere. So even in microscopy, unless you're following individual axons, as soon as you have a pixel that can have multiple axons in the pixel, you have crossing fibers everywhere. So that's really a problem for connectivity. And I don't know if Obai or you or some speakers will talk about this week, but actually if you try to, I'm going to say maybe something wrong here, but if you try to not explain, but yeah, see how the white matter supports the functional MRI data or the EEG data. If these connections are missing, these things that are, you don't explain the data well. And at the time where people couldn't do good tractography, I remember there's a paper that actually people started manually adding connections that we knew existed from anatomy, right? So they took the connectivity matrix and they said, what? Tractography doesn't give me these connections? Like I know these two parts of the brain are connected. Why are they not connected in my matrix? Well, let's put a 1 there. And let's put a 1 there. 1, 1, 1. And suddenly, ooh, it explains the data much better than if their connections are not there. So that's when we started working with Abid and his group saying, look, maybe that's a way to evaluate the quality of my tractography. Is a better tractography explain better the functional data. And I think this is something you're going to hear about in the coming weeks, I guess. Maybe not.
Yeah, Obai is going to talk.
Good. So all that to say that before we started modeling tractography well, all these years that people were doing DTI tractography, to me, they just had in their hands a low order approximation of the white matter. It's not that it was wrong. It's just that they were missing a lot of connections. A lot of true negatives were missing. False negatives. And so, and this was quite, well, it's a bit small, but if you start going beyond the single tensor and suddenly you allow yourself to have or two tensors, one tensor here and one tensor here. Not even talking about interpreting microstructure, but just for tractography, this is critical because suddenly you're coming from the corpus callosum, and you can see here I have a red peak, red, and I'm traversing the green, and boom, I can actually start connecting left and right hemisphere. With DTI, and I'll show you an example, I was just getting this. As soon as I was getting here, it's like, I don't know where to go. And you're pushed towards the mean, and you go up. Impossible to traverse this traffic jam. These hot spots of low FA. You see behind I put FA. These are all locations where the tensor is a disk or even a sphere. When you have three-way crossings here, this is actually a sphere. It's a perfect XYZ track crossing. Yeah, maybe I can fast track. Well, yeah. No, you'll see it later. And so that was the three of us in 2007. Yeah. So at the same time, as a student, that's super stressful. But looking back now with maturity, it's like, okay, we needed this invention. Back then, I was fighting to get my paper in because I was two other groups at the same time were coming up with the same ideas and the same techniques. And looking back is like, we were on the right track and we all differently thought about this solution. But nowadays, this notion of crossing fiber modeling, no matter what tool you use, is it spherical deconvolution or multi-tensor fitting or whatnot, the essential here is that it's critical to acquire good images, but also to reconstruct the multi-orientational features of the signal at every voxel. And this is not necessarily for such longer acquisition. It doesn't come for free, right? DTI was on the order of 3 minutes. Today, we're on the order of 5 to 10 minutes, but so you need a bit more data. Of course, suddenly you have 2 or 3 tensors. So instead of just 6 unknowns, you have 15 to 20 unknowns to fit. The images are still noisy, and as a bonus, you would want to be a little bit more specific to intracellular, extracellular water. So there's no free lunch. This comes at a price, and typically nowadays it's a 10-minute acquisition. When we have clinicians that say 10 minutes is too long, I cannot keep my patient in for 10 minutes. Les fantômes du CRM. So we can work it out in 5 minutes. Great. So you're going to go home today with a new word, I think. Pixel is a picture element. I think all of you knew it. 2D pixel. Voxel, you knew that one. What's a pixel? Same logic. Now we're modeling crossing fibers, so we need a new term. Yes, good. I rarely get it in the audience. Suddenly we have a fiber element concept. This was introduced in 2012 or so. It's starting to percolate and make its appearance in papers. But it's this concept that within the voxel, now we're going to have pixels, right? This would be pixel 1. This would be pixel 2. In theory, this is a 5-dimensional space. So we have as many pixels as theta phi, as angles in the voxel. So this can be a computational nightmare or something we can discretize on a few main pixels in each voxel. So suddenly we're talking about pixel analysis. So we have a whole brain tractogram. I'm going to show you next how we can start dissecting this tractogram virtually to extract large family of bundles. We're going to zoom in into a voxel. We're going to have a pixel for the blue track. We're going to have a pixel for the orange track. We're going to have extracellular water. And suddenly our microscope, right, our virtual microscope at the macroscopic level is starting to be a bit more specific. So each track, right, this can be a vision track or motor track. It might have a different microstructure than the orange track. So we need speed of water for this track versus speed of water for this track. The amount of axons here, the intracellular water here can be different from this track. And that's the new holy grail of tractography. And this afternoon, that's actually, I don't know if the speaker is in the room. Is he in the room? No? But he's going to talk about G ratio. So mapping the G ratio is mapping the ratio between intra and myelin across the whole brain. Can we do this across tracks? And I'm going to talk about it a little bit in I'm going to introduce what you need to understand this talk, but now you can start imagining that at least locally, if we have enough data and our models are expressive enough, for each pixel, we can have 3 numbers. The amount of axon, and the amount of myelin, and the amount of water outside these structures. 3 numbers per pixel, right? And then 3 number for this pixel, so that's 6, and maybe 1 number for the amount of water overall in the voxel. So give this to a clinician. How do you give this to a clinician? We're not there yet, right? But suddenly, right, this is the famous arcuate fasciculus, the language tract coming here in the temporal lobe. Suddenly, instead of following FA, right, I chose 3 pixel, 1, 2, 3 voxels, and the FA there is perfectly equal, 0.4. Same diffusion tensor. But look at the different orientations and different profiles. So suddenly I can start following the FA or the axial diffusivity of the track, right? It's going to be green, blue, green. And instead of seeing dips whenever there's a crossing, I'm really going to start following the microstructure of that track. And here I used apparent fiber density. The A, like apparent diffusion coefficient, appears. So the apparent fiber density for each peak in the voxel. So the green one and the red one. You can see the potential. So if suddenly we have disease, and that was my tentative here, 1 fascicle is healthy, but just the second one is deteriorating. This is troubling a lot of DTI people, but you can actually see that this tensor here in the patient is actually more anisotropic. This is FA increasing with disease. Like, what? How do I publish this result? Let's not publish. No, let's go see underneath what's happening. Underneath, what's happening is that the blue and the red peak are the same, but the green peak is changing. So what I tell my students or collaborators always is, okay, start with DTI. FA is dropping or FA is increasing. What's happening? Is the number of fiber orientation changing? Yes, no. No. Are you in a crossing fiber voxel? Yes. Oops. Don't trust FA. Let's go see and what's happening in the pixel space. That's a way to try to interpret a little bit more specifically. Tractography. You guys tired? I'm getting tired. All right, that's the easy part in a sense. We're just going to connect the dots. But that's where also it's. First, we need to start from somewhere, and we need a domain for tractography. Okay, and so in all tractography algorithms, you need a seeding mask. Where do you start? Where do you seed? So if you're working with surgery, it may not be the same solution as you're working with inferring functional connectivity in the brain. But typically, we seed the whole white matter. That's usually the default in most algorithms. But more recently, especially if you're going to do cortical mapping, correlation with the surface, with fMRI, with EEG, and because our segmentation algorithms are becoming better and better, a lot of people for connectomics go for this approach. Seeding from the gray matter, white matter interface. Or even better, using the surface. So that's something, a technology we introduced in my lab and that we brought to Obay and Habib, but we can actually use the surface of the cortex and the nucleus and the normal to this surface to start tractography. Then tractography is super naive. Once you have a seed, basically you just need a way to discretize space and follow pixels or follow DTI, principal eigenvector. That's the most naive approach. You start at a point Pn. You have a vector at this point, right? V. This is a 3D vector. You take it times a step size. You're at the new point. You have a vector here. So of course you have to interpolate. And you step. And you step. And you step. And you walk in white matter like that from every seed to everywhere else until you meet a stopping criteria. I need to accelerate a little bit. And so that's an animation. Here, in fact, this is a functional MRI seeding strategy. You'll see the blobs later. But I actually started from a motor task fMRI activation. I'm going to play it again. You can see it's dirty also. Now I'm moving backwards. But this is just putting a seed at every point at the beginning and just following the pixels until I step out of white matter, more or less. But inside the algorithm, and this is why it's important, especially if you go to microscopy or macaque or mouse, and suddenly these notions of step are smaller because the pixels are smaller, you need to adjust the parameters. Because remember that all our parameters are fixed for typical 1 by 1 by 1 millimeter MRI data. But all of these are options. How do you step in white matter? Luckily, we've been doing this for almost 15 years now, so we have good priors on what's a good step size, what's a good theta, based on the data you're providing. So a recommendation, of course, this is a star. I mentioned this earlier, but if you have DTI only, you just have this, the curve, like the mohawk. And suddenly with crossing fibers, you get the whole motor homunculus. You can traverse the traffic jams. But as we're going to see after the break, it's also easier to get lost. A recommendation nowadays is to use probabilistic tractography. So instead of picking always the principal vector, the V, you actually sample your data to exploit the variability and the in the continuous modeling, and so you can get a bit more spatial coverage without losing yourself. So this allows to traverse white matter better. So most software now have probabilistic tractography as a default. FSL, MRTrix, DIPI, us, and TractoFlow. And you can see these are papers that are getting old in a sense, and there's a consensus here in the community. So let me just fast track this. And so using your crossing fibers, using your masks and your seeds, you're able to reconstruct millions of lines. And in about 10 minutes. So quite fast. And of course, if you follow my recommendation, the quality of this tractogram will be better than if you use DTI. You're going to get through crossings. You might get a bit lost. We're going to talk about that in the second talk, but you're going to reach gray matter and you can start actually creating a connectivity matrix here, right? You can actually start wondering, how are these 2 points connected? How are these 2 points connected? First, are they connected? Maybe the first question is binary, 0, 1. And then unfortunately, because this was invented 20 years ago, network neuroscience, at the beginning, people all they could use is, okay, yes, no, they're connected. Then what do I put in the matrix? And people put the average FA in the matrix. See, that's not great. I said all models are useful, some are wrong. This I just don't understand why people started doing that. But they did. So there's at least 10, 15 years of publication in network neuroscience where people tried mean FA between pairs of points. Don't do that. I think that's a bit out. Took us another 10 years to kind of clean that up. But what's still out there, and we're going to see this afternoon, we can do better, is what else could people try than put the number of lines between 2 points? We're going to see this is not perfect, but most people now use how many lines, how many tracks are there between A and B, B and C, and they build a matrix this way. Let me just see where I'm at, because we're not going to finish. But I started 10 minutes late. Can we have 10 minutes? That's what, and then I can cut on the research side, but okay. Lots of tools produce this tractogram and they say, okay, bye, we're done. Good luck. What do I do with this? You know, 10 gigabytes of lines and a TRK file, a track file. What do I do with this? What can I do with this? Two applications in my mind. One is tractometry, try to quantify tracks. And the other is connectivity, connectomics. So luckily now we have tons of tools that can start dissecting this or segmenting this with rules, anatomical rules and shape of lines to extract the famous highways of the brain, the corpus callosum, the cortical spinal tract, the fornix, very important for memory, the superior longitudinal fasciculus, the ILF, the uncinate. So again, this is it's nice to see that the field is moving forward. There are many methods out there, but all of us as a community are converging on 50 or so tracks that are almost press button. So you run TractoFlow, you run MRTrix, and then you feed that to a segmentation algorithm, a little bit like FreeSurfer, but this will extract tracks, regions of interest. And so with our tool here, and that's included in our pipelines, you have more or less 50 such tracks that are robust to crossing fibers. You can see here they're quite full. They're not the typical tracks you see in the papers because they're rich. They're really starting from large cortical areas, traversing crossing fibers and reaching the cortex. And so one way to use DTI, tractography, and diffusion in your study is to actually start doing tractometry. quantifying whatever in these tracks. And I haven't seen a lot of even simple ideas of let's see how the functional signal behaves at the endpoints of these tracks. What is the bold correlation there? What is my myelin content underneath? What is my quantitative susceptibility maps there? There's so much you can do here. For me, this is the backbone of anatomy. You have now white matter tracks that you can use like a region of interest for anything. So for instance, in Alzheimer's disease, we always go to the fornix, the cingulum bundle, part of the corpus callosum. So now we can run a whole pipeline, extract these features and start quantifying them with disease, with aging, with neurodevelopment, with MS, with Parkinson's disease. And so these tracks of interest change depending on your application, depending on your disease, depending on your project. And so this is really possible at the moment. And so what is tractometry? Well, it's the concept of metry, so measuring something in a track. So given this cortical spinal tract, for instance, whoops, there's lots of ways you can quantify it. First, you can measure its volume. This is general shape. So we have macroscopic features. We can measure the area of cortex. We can measure the area of brainstem, the size. This we start seeing in the literature. But what is most important is to try to quantify the microstructure. So what is the FA? What is the axial diffusivity? What is the fixal axial diffusivity? And so everything I introduced now since the beginning can be applied here because suddenly you have your track of interest. So in a sense, you have your fixal, right? For every voxel here, this is the mask of every voxel traversed by this track. In a sense, inside these voxel, you have your fixal of interest now, given by this track, this orientation. So you can have the apparent diffusion coefficient of this track. Robust to crossing fibers. You can actually follow it in the signal. And then create signatures. This is called track profiling. So from tail to head or head to tail, you can actually follow a certain metric, whether it's FA or fixal-based something along the track, and suddenly you have a signature. What is this is kind of the fingerprint of this track. And that's becoming very powerful because imagine a nice multimodality acquisition like we can do here at McGill or Concordia and Sherbrooke, where you have a myelin contrast, you have diffusion, you have fMRI, you have FLAIR, you have QSM, and suddenly you can create a fingerprint like that for all your modalities and start building this into a classifier or whatnot, depending on your research question. But it's hard because it's very high dimensional. We have 50 such tracks. Each track has 10 or more features. So yeah, we need new statistics. We need new computational methods and models to analyze these inputs, these not new inputs that now we trust. And so we can do that for every track. For every track, we can actually separate it in 5 to 50 sections and have fingerprints of these tracks with Alzheimer's disease and whatnot. And I think this is a very also hot topic for medicine, and with the company we build, we're starting to bring this to the market and to the clinic, a little bit like when we bring our kid to the hospital, to the pediatrics, and we measure them and we put them on a curve. Now we're building normative references from 20 years old to 90 years old of what is a healthy white matter. So that if you have a concussion, a stroke, or you show up to the ER and you're this red curve, we can actually map where you are. what is the condition of your white matter with respect to a normative reference? And so thanks to the huge open projects out there, now we have more and more data, more and more tools to pool data together, because we said, you know, these data sets are very variable across sites, but this is now imaginable. And last but not least, but just 5 minutes on the connectome. We have pipelines. Just little publicity for the lab, but it's called the free surfer flow, but that's a bad name. But we have a flow that can basically generate all the parcellations you like in diffusion space that fit well with tractography. A lot of these atlases don't fit well with tractography. They don't touch the white matter, they're just the cortex. So we need to be careful and register them properly. And then we launch ConnectoFlow. And essentially, we're taking the T1, the diffusion, we're doing TractoFlow for the tractography. We're using FreeSurfer or Lausanne or Schaefer or Brainatome, or I forget how many we have, but we have like 12 of them out there, which is just a way to parcellate the brain. But most importantly, to make it tractography friendly. So we're using the surface of the cortex and the endpoints of tractography to make sure they connect well, and we build the connectome. And the connectomes, because we don't build the FA connectome, but you can basically build the connectome for every pixel measures that I presented today. So not just streamline count or track count, but axial diffusivity, extracellular space. You can even put lesions in there. So it's really a pipeline to handle connectomics data. And just to make sure everybody follows, you know, between two parcels here, internally, we have all these tracks, so we have all the pixels. And so we can basically put any number here that we want. We could put the volume of the track count, the cortex touch, the FA if we want, the axial diffusivity, whatever we want in this. So it's actually monstrous. The pipeline is really made for data science because it produces, I think, 25 connectomes of different features of the white matter. We have lots of strategies to deal with problems of tractography, and we have ways to visualize it. So this can be quite useful. You can visualize, interact. We have a SIL connectome math where you can play with the mathematics of the connectome, and you can extract also your graph theory features of the connectome. So in conclusion, well, if you go back to the video, I think you'll be convinced that you need to check your data, use the tools out there to create HTML reports, inspect your data, use crossing fiber modeling before tractography. And if you start quantifying the white matter, use pixel-based information. Be careful of the press button solutions. I say that, but I'm creating tools to press a button. But I try to teach you and give you the tools to actually look under the hood and adapt it. That's why there's a warning here. There's like, this is not perfect. It's work in progress, but it's definitely usable. And I would love to see more white matter integrated in neuroscience and neuroimaging in general. So yeah, let's break here. But keep in mind that if you have pathology, most of these tools will break or will need to be adapted, and we can talk about this after the break. All right, thank you.